Okay, so today we're going to take a whirlwind, everything is a whirlwind tour. So we're going to take a whirlwind tour of bats. So you might wonder, like, why, is bats relevant to cognitive science? And it turns out that so there's a lot of study of bats, bat echolocation, and it turns out that certain aspects of the signal processing involved in the way the bats process their calls and their echoes are surprisingly similar to problems that you have to solve in speech perception. And the bat calls actually look a little bit like consonant vowel or vowel consonant syllables. And so given that you've got the same raw material of the auditory system, you got the nucleus of the lateral lemniscus, that's present in bats. The kind of strategies that you end up with are probably somewhat similar. And so bats have been studied in great detail, just a small number of bats. So not all bats echolocate. So you might have seen, if you've been to San Diego Zoo, from time to time they have fruit bats, and they've got these like really big thumbs, and they sort of hang onto the cage with their thumbs, and they eat. They're sort of like up at sort of dawn and dusk. Those guys don't echolocate. So not all bats echolocate. So there's the mega bats don't echolocate, but the small bats, some of them do, and they use different systems. And so one of them, the little brown bat, has been studied in a lot of detail. And so this isn't, what I'm going to tell you, isn't true even of all bats. They sort of, some of them do it different ways. But we always have to have a little bit of evolution. So, where do bats come from? So here's 0 million years, the present. And here's 60 million years ago. And there's a big meteorite that hit in the Yucatan. And so basically before that, dinosaurs were basically in control of all the daytime real estate. And the only dinosaurs that made it through were the birds. So basically birds are, the descendants of the dinosaurs that survived the meteorite. And I always like to mention, if you go back to say 200 and I think it's 230 million years ago, that was the Permian extinction. And that was the, Mother Earth sometimes is a little harsh. And so during the Permian extinction, 90% of all life went extinct. So that was the really big one. This extinction wasn't as big, but because, and so when you actually look at, what replaced the dinosaurs, it was all these mammals radiated into all these open niches, and so that's how you got all the sort of modern mammals. But the birds were still occupying the daytime real estate, and so that's why. So here's the night. So that's why bats are at night. And bats are the most common mammal because most birds don't fly around at night, and so bats had some open real estate to colonize. So bats are very small mammals, very small brain, smallest sort of brain body weight ratio of any mammal. They're also the most common mammal. There's more bats than any other kind of mammal. And what do? Well, they eat bugs. So obviously that doesn't have anything to do with speech perception, but it turns out they use these signals that are sort of somewhat speech-like. And so they're really good at eating bugs. And why is that? Because they're so small. And so the reason they have to eat their body weight in bugs every day is because if you look at their surface area to volume, so if you take a cube and sort of double the size of a cube, you'll find that the volume increases much faster than the surface area. And so the reverse is true. So if you shrink a cube down, you end up with a lot of surface area and not too much mass. And so they'll freeze to death if they don't eat their weight in bugs every day. So they're very good bug eaters. And so how do they do this? So let's draw a bat here. So here's the bat ears. So they got big ears, and then they got there's the bat mouth, little tiny eye. And so what they do is they send out an extremely loud, they have extremely loud voices. And so they send out, so here's a little fly here that they're looking to eat. So they send out these calls. They're like 120 decibels. That's like the sound of a freight train, but very high-pitched, ultrasonic. And then they get a tiny little echo back from that bug, which they then analyze. So a bug, you can imagine a bug doesn't make much of an echo, but and you might wonder, like, if their voice is so loud, how do they actually deal with their very sensitive ears? And they have extremely fast muscles in their ears, and so what they do is they grit their ears, and then they go bonk and send out this sound, and then they quickly let go of the muscle in their ear on the stapes, and then in time to actually sort of get the echo. So that's how they actually manage to have such a loud voice and have these very sensitive ears. So if you look at the bat, if you look at the bat vocal cords, so one of the things we're going to have to talk about, I talked about it in the advanced session, but I'll talk about it again here. The vocal cords generate harmonics, and so what does that mean? So if you take if you take the low E on a guitar, I think it's like 82 hertz, and if you pluck a guitar string, it's battened down at either side, and it will sort of vibrate back and forth. And what do you call that? You call that the first harmonic of the guitar string. And then you have integer harmonics. So depending on where you pluck the string, you might cause the string to do something like this. Or you could also have a combination of those two added onto each other. And so that's the second harmonic. So the string is going back and forth like at 2, the second harmonic. And then you got the third harmonic, which is, they're always integer, multiples, because of the fact that the string is battened down. But it turns out it goes way up. You can go up to the 14th harmonic. So what's the frequency of these harmonics? So if you look at, say, we'll just round to 80. So if the low E on a guitar was 80 hertz, this guy is going to be 160, and this guy is going to be 240. So basically, these are just integer multiples of the frequency of the first harmonic. So that's happening in a bat. So why is this relevant? Well, how do the vocal cords work? They're basically like little stiff rubber bands, and they're tensioned up, and then you blow air through them with your lungs. And so what happens is they vibrate, and so they vibrate in all these different harmonics, like up to the 14th or the 18th harmonic. So what we want to do first is just look at what the actual bat call looks like. Now, like I said, this is just the little brown bat. There's other bats that have different ways of doing it. But if we plot out the call here on a good-sized graph, I'll leave this one up. So what's the first harmonic of the bat? So they're little tiny bats, and they got little tiny vocal cords, and so it's really high-pitched. And so the first harmonic is about 30 kilohertz. So that's higher than you can actually hear. And so this is 30 kilohertz, 30,000 hertz. So young people out there, you can probably hear 20 kilohertz still. I'd be lucky if I can hear like 9 kilohertz. So 30 kilohertz is, is generally beyond the range of human hearing. And so they send out a short constant frequency part, and then they roll off and make like a little sort of bend at the end. That's called the frequency modulated part. So this guy is called the CF part for constant frequency. And this guy is the FM part for frequency modulated. So that's the first harmonic. So that's equivalent to this guy. But then you're going to get integer multiples of that. And so there's going to be a second harmonic, and it's going to be at 60. That's a really loud one. It's going to be at 60 kilohertz. And then there'll be another one at 90 kilohertz. They can actually still hear that. And they can even hear a little bit at 120 kilohertz. So what are we plotting here? So this is frequency going this way and time going this way. And what kind of time are we talking about? This might be, say, say 50 milliseconds. So that's only a 20th of a second. So this is like it's a very rapid with a little bend at the end. So that's basically what goes out. So this guy is the outgoing. So constant frequency and outgoing. So outgoing is a thick line, and the echo is dotted. So then we get an echo back, and so what's going to happen with the echo? The echo is going to be several things. I'm a little exaggerating the amount of shift here, but the echo is going to come back not at the same frequency. And so why is this? So why does the echo not come back at the same frequency? So to understand this, we have to talk about the Doppler shift. So there's two things about the echo. So one is it's going to come back at a different frequency, and then it's also going to come back at a different time. There's going to be a delay. So there's going to be, a change in frequency here, and there's also going to be a change in time. So what causes that Doppler shift? So, and it turns out the bats have to measure this. This is important for bats. So Doppler shift. So a Doppler shift, the classic example of a Doppler shift is a train comes by. 19th century train comes by, and it goes, when the train is coming toward you, it's got a higher pitch. And then when it goes by, it just goes, it sort of like goes down when it goes by. Now what happens if you're on the train? Well, it's just going the whole time. So what causes the Doppler shift? So here's the train coming toward you. Here's the person sort of listening to the train. There's their ears. So the train's going that way, and some sound waves are coming out of it, but the source is moving. So basically what happens is, because the source is moving, those sound waves are going to hit your ear more rapidly than they would if the sound source was stationary. And so in this case, the Doppler shift is up. So the pitch. So when the train's in a distance coming toward you, it's all the pitch is elevated. Then it passes you. It's a little complicated geometrically when it passes you, but we'll just let it pass you. And then now the train's past you, going away from you, it's still emitting sound. And so the sound waves that come out of the train afterwards are on a source that's moving away from you, and so they're going to actually hit your ear at a lower frequency than so this is Doppler down, because the source is moving that way. The same spacing of waves come out, but if the source is moving, they're going to hit you. So that's basically the Doppler shift. It just changes the frequency depending on what the relative velocity is. So it's the relative velocity. It's the velocity. You could be moving too. That would affect it, but it's the velocity difference between the source and the thing that's listening. So why is this relevant? So if you look at it at a bat, and if you look at the whole sort of way that bats, how do bats work? What they've done amazingly is they've created an acoustic analog of the fovea. So why do we have the fovea, which is the high density part of the retina? We have it so that we've got some really high resolution vision right in the middle, and then we lower the resolution so that we can essentially detect very fine details, but not have an optic nerve that's so gigantic if we tried to sort of service the whole retina with that same high density at the middle. So, and then we've got a system for eye movements to move that sensitive part of the retina around to look at detailed things. So bats have done the same thing functionally in the auditory system using completely different mechanism. And so how does it work in the retina? In the retina, you just cram all the receptors, the photoreceptors, together, so you get like a really high density of photoreceptors, and then you kind of swish out all the ganglion cells out of the way so that the light can come through without hitting them. And so it was possible in the retina to actually change the density of the receptors and have them still work. You can't do that in the auditory system very easily because hair cells are like a certain size. You can't sort of shrink them down very easily. And so what the bats have done instead is they've actually taken the stiffness of the basilar membrane and flattened it out. So it looks like, if you look at their basilar membrane stiffness, it looks something like that. So this direction is stiffness, basilar membrane as a function of where you are on the cochlea, and then this is distance along cochlea. So they couldn't jam the receptors closer together, and so what they did was they just flattened out the gradient of stiffness. And so what this does is, this is about the amount of stiffness that you need for 60 kilohertz. And then this is the amount of stiffness you need for just like 62 kilohertz. And then, it gets stiffer and stiffer. The stiffer it is, the higher the frequency the basilar membrane vibrates at. So this gets up to like almost 120 kilohertz up here. And then, this is like, say, 10 kilohertz here. So what they've done is they've created a fovea. They've created an extreme sensitivity to sounds between 60 and 62 kilohertz. That's like a fovea. And then what they do, how do they sort of what's the equivalent of an eye movement? So they have something called the Doppler shift compensation behavior. So what they do is they actually send out an echo. They send out a call, the outgoing. So this guy, that's outgoing, versus echo. So they send out the outgoing. They get an echo back, analyze it, figure out what the Doppler shift is, and then they adjust their voice so that they get an echo that comes back in this range. So they're kind of doing this something, it's kind of equivalent to an eye movement. It's essentially doing something with your motor system that will cause the signal to come back to a sensitive part of your receptor array. And so, so this is the acoustic. So what is this? It's detect Doppler, and then adjust outgoing to get 60 to 63 kilohertz echo. And so they definitely have to measure the Doppler shift. And so there's a lot of work on this, and I put some readings. A lot of this work was done by Nobuo Suga. And there's if you want to look up this a lot of this very interesting research. So that's the basic plan of the bat. And what does it need to get out of the echo? Well, it needs to get a bunch of different things. And so one of the things it needs to get out of the echo is the relative velocity, because it wants to figure out how fast the bug is flying away. Is it flying away, or is it flying toward you? Or is it a wall? Maybe there's a wall there. You don't want to fly right into the wall. It's not a bug. But then you also want to know the distance. That's different. How far away it is. And then, of course, then you also need, what is it? Is it a wall? Is it a bug? Is it is it a branch? So bats can basically see the world with this echolocation system. And I always tell the story. I walked into a bat lab once, and there was a little bat. It was in his little cozy little bat box. And his not very good vision realized, that isn't the experimenter. Who is that? So the bat sort of got out of the box and looked at me, and it just went, just turned on the radar. I couldn't hear it. It was just checking me out, who are you? What do you look like? And so bats are. Another story I tell is they can detect. If you put some wires in a room, completely pitch black room, they can fly around the wires, so they can actually detect tiny echoes off of a wire, and navigate around it. And they're not stupid. They remember where the wires are. So if you move the wires, they'll run into the wires because they didn't have the radar on. They weren't paying too much attention. So they basically can sort of completely perceive the world through the auditory system without any vision at all. So we're not even scratching the surface of how they do that. We're talking mainly about sort of a few relatively simple things they do. How do they actually sort of determine it's a wall versus a wire? There's less, information, on that. Or whether it's a good bug to eat. Maybe it's a poisonous one. They don't want that one. Okay, so that's the bat picture, and there's a lot of work has been done looking at these different auditory areas, and I'll just talk mainly about one of them, or two of them. So if you look in a bat, here's a little bat brain like that. There's the olfactory bulb. They got a cerebellum. There's the fourth ventricle. So if you look in a bat, there's, they got really quite a big auditory cortex. And then there's several other areas sort of near it. And if we blow those up, we get the primary auditory cortex. And the primary auditory cortex, it has this big sort of bullseye-like thing in the middle of it. And that's pretty much the representation of this very sensitive part from 60 to 60 kilohertz. And it has this sort of strange sort of bullseye-like structure to it, and where it's like 61 in the middle, say 62, 63 out here. And then when you actually move in this direction, it actually maps the amplitude. And that's where the sensitive echoes are being analyzed. We won't talk about that too much. But then when you get away from that really sensitive sort of acoustic fovea part, then you have other frequencies out here. So this is like 10, 20, 40, 60, like that. And then it continues on like up to like say 110, 90, 70 here. So this is basically primary auditory cortex. So this is A1. But the areas that we're going to talk about are these areas that are sort of sitting up here. So there's several additional areas that are in secondary auditory cortex, and there's another sort of big one that sort of comes over here like that. It's got several subdivisions in it. And the one in particular we're going to talk about is this one called the constant frequency area. So this is constant frequency 1 / constant frequency 3. It's one of the Doppler shift areas. And these areas are frequency modulated areas. So those are frequency modulated 1 - frequency modulated 2. And there's some other ones up there, and there's a couple of them. I'll just talk about one. So that's a higher auditory area. And when people looked at it, they discovered it was actually an area that had neurons in it that are sensitive to more than one frequency. So here's the CF. So I'll draw out that area, that CF1, CF3 area. So if you draw it out bigger, what does it look like? So, like I say, it's like everything in this class. It's, what do you got? You got cylinders, you got squares. So let's draw this area out as a nice square. And it turns out it has neurons in it that require 2 frequencies at the same time in order for the neuron to fire. So basically, if you have along one axis of this area, you're going from 30 to 32 kilohertz. So here's 31 kilohertz. And then on the other axis, you're going from 90 up to what you would expect for the harmonic of 32, which would be 96, because it's going to be 3 times. It's the third harmonic, so it's going to be 3 times the frequency. So this middle would be 93. That corresponds to 31. So the way this area works is if you play in, say, 31 and 93, a neuron at this location in the area, so we'll make this little 3D thing here. So this is like a little patch of the cortex, okay? A neuron at that location will fire. But if you play in just 93, it won't fire. If you play in just 31, it won't fire. You got to have both. And so there's a map of these double frequency neurons that require two frequencies in order for them to fire. And so now let's look at what the coordinates of this map mean. So if you're down the diagonal of this map, what does that mean? So if you're down the diagonal of this map, that means you like 30 and 90, and 31 and 93, and 31 and a half, and 94 and a half, and 32 and 96. And what does that correspond to? That corresponds to no Doppler shift. So what is this area doing? It's actually sort of comparing a constant frequency part here to an echo up here. So this is, this was 30 kilohertz, this was 60, 90, 120. So it's not comparing, it's comparing an echo from a different harmonic to the outgoing of the first harmonic. That's what this area does. And if it finds 30 and 90, or 96 and 32, that's along the diagonal of this cortical map. That means no Doppler shift. So this is Doppler shift equals 0. Now, if you're off the diagonal, then you're going to have a negative Doppler shift. So if we're over here, or over here, this is this is negative Doppler, because like what happens for a neuron right here? So this neuron likes 31 and 91, but 31, if there was no Doppler shift, should be 93. So the echo came back at a lower than you were expecting frequency. So that would be like a Doppler shift that was actually underneath the outgoing. So that's a negative Doppler shift. But diagonals along here are going to be, positive Doppler shifts. So this is going to be, positive Doppler. So what do you have in this cortical area? So you've got a cortical area that maps, two frequencies. But if we look at it, if we just move in a diagonal direction, what is that? That's basically the Doppler direction. So that's the Doppler shift direction. And then what's this direction? This direction here, that direction is different voice pitches. So this is, different, outgoing. Because you can see what the problem is. If the bat is adjusting its voice all up and down, then it has to calculate the Doppler shift, but it's not a specific frequency. What it really needs to calculate is, what is the frequency difference between this outgoing and this echo? But it needs to be independent of frequency, because this frequency that the bat is sending out is going up and down all the time because it's constantly adjusting its outgoing. And so what it needs to do, this is sort of getting down to what is the fundamental signal processing thing it has to do. It has to detect the difference between two frequencies, the spacing between two frequencies, independent of where they are. So it needs to detect, like, is it this, or is it slightly this that's further apart, or is it this, which is closer together? That's what the computational problem is. And so how could it do that with this map? Well, if it sets up this map, because the nervous system likes to do maps, if it sets up this map, then it's relatively straightforward to extract that signal difference out of this map. All you got to do is just sum across a diagonal. And when you think about this, it's sort of, if you went into this system, we said that, you tend to get these projections that are sort of plane to plane, like, a plane in one area will project to, a plane in another area. That's what happens in what tends to happen in the auditory system. It'll sort of like project to a plane. How could you do this kind of signal processing thing? Well, just tweak this, just tilt this plane a little bit. If you tilt this plane a little bit so that it's sort of like at 45 degrees, then if you looked at it, you would say, it just looks like a regular point-to-plane projection. But what you've done is you're summing, instead of summing across isofrequency plane, you're summing diagonally across an iso-Doppler shift plane. And so it's actually not that hard when you think about it. It's actually just a little tweak. All you got to do is just like somehow tell the developmental neurons, just rotate your plane a little bit, and then we'll sum across a plane of iso-Doppler shift. And so then it can solve this problem. And the problem is, try to find the distance between these two frequency bands independent of what the absolute frequency is. That's really what the what the computational problem is. So what does this have to do with speech? It doesn't sound like speech at all. So now we have to sort of have like an introduction to acoustic phonetics in just about 10 minutes, but there's not too much to it. It's sort of basic, sensible thing. So how does speech sounds work? So we have to draw an ugly head. For some reason, I cannot improve my attractiveness of my head drawing. So here's a person, and there's their mouth. So their mouth is open a little bit. So if you look at that person there, so how does speech fundamentally work? Well, you've got a big tongue in there. Most people don't realize how big the tongue. If you take an MRI, you realize, dang, the tongue is a giant thing. It goes way down into the throat. So here's the tongue. You got this giant tongue. You just see the little part that's sticking out like that, but it's an enormous thing. It goes all the way down into the throat. And what that tongue can do is it can control these two different cavities. So if you look, there's an oral cavity, and then it sort of necks down a little bit, and then there's a pharyngeal cavity, and then at the bottom is the larynx. So that's the larynx that makes noise. But there's these two. So this is the filled-in tongue, just a giant muscle. So there's two separate cavities here. So there's this guy's the oral cavity, and this is the pharyngeal. So this is oral and pharyngeal. Cavity, and those can be independently modulated. And how do you do that? Well, you can move your tongue up and keep the back of the tongue about the same, or you can move your tongue to the back and keep the spacing of the top oral cavity, or you can change both at the same time. And so this guy is the larynx down here. Yeah, so that's the larynx that actually makes the sound, and this is a very then it's attached up to the lungs, but this turns out to be a relatively dangerous setup. And so most animals, the larynx is up here. And why is it up there? Well, it's up there so that when we want to attach the mouth up to the gut, we won't accidentally get food down into the lungs. And so what happens in babies, they're born with a normal mammalian or even turtle larynx that's up at the back of the throat where it should be, not down. And then what happens in the first couple months of life, as the tongue grows, it kind of pushes the larynx down into the throat, into the typically human position. And of course, there's more choking problems in humans because of this, because there's more of a chance of getting some food down into your lungs or blocking the whole thing. But that allows us to generate all these different featured sounds. And so now we have to talk about, like, what do the sounds actually look like? What are the sounds that come out of here? So it's not exactly the same as the bat. So what happens is you can think of these cavities here as a filter. So you can think of the larynx as producing zillions of harmonics, like 16th, 18th harmonic, all integer multiples of the pitch of the voice, and then you run them through this filter, and this filter has two adjustable resonant cavities, and those can essentially generate bands of a particular frequency. And so if we draw out a graph of what a vowel looks like, so here's a graph of a vowel. So let's just say we were doing something like E, or just going up. So E, so it sounds like an E, even though the voice pitch is changing. So here's the voice pitch. So say I start off at 100, and I go up an octave like E. So that's so that means it's 200 over here, and it was 100 over here. So that's the first it used to be here, the first harmonic of my vocal cords. But then I've got the second harmonic, and I got the third harmonic, and the fourth harmonic, and it keeps going. It goes up to, if you look at a spectrogram, you see it goes up to like, harmonic 17, 18 goes. So each one is integer multiples. So that's what's coming out of the larynx. And how do you figure this out? You can just take a tiny little microphone, stick it right down right next to the larynx, don't cough, and record, and you can see all these harmonics coming out. Then it goes through these filter cavities, and what the filter cavities do is they emphasize a particular band. So maybe they emphasize this band, they let that frequency through, or they let this frequency through. And so what that's going to do is that's going to create, like, whatever harmonic happens to be at this frequency band will come through. And whatever harmonic happens to be at this frequency band could be a different harmonic. So it's different than the bat. So in the bat case, these are the actual harmonics. Here, these things are just emphasized bands of whatever the hell harmonic happened to be at that frequency. And so this thing is called, this is called F2. That's called a formant. That's the phonetician's name for this emphasized band. And here's F1. And then they sort of inconsistently call the fundamental frequency F0. It's kind of confusing because the first harmonic should be the first harmonic as in 1. But in speech, they call that F0. That's a different thing than this. This is this emphasized band. But that's basically what allows a vowel to sound the same no matter what the voice pitch is. Because I can go like E, it sounds like E, and then I can go ah. My voice is going up and down. My voice pitch is going up and down, but the sound sounds the same because my tongue hasn't moved. And so what's the problem? Well, people have different sized vocal tracts. So if it was the case, I could say like the formant 1 should always be 800, and then I can tell that this is an E because there's a relatively wide spacing between formant 2 and formant 1. And if I said an ah, this formant 2, because I changed where my tongue was, this formant 2 would come down. So all I would have to do is just look for 800 and 2000, and then I would be done. But unfortunately, that doesn't work. And the reason it doesn't work is because people have different sized mouths, a lot different sized mouths. Like a baby has a very small mouth. And so what does that do? That does something a little bit like the bat. So it causes all the formants to shift up. So if you actually look at, if we make a graph of, let's make a graph here of formant, say formant 2 versus formant 1, and then have somebody say an E. So maybe that will be one person's E. But a person with a small vocal tract, E, might be up here. And so what will happen is there'll be a whole sort of smear of different pairs of frequencies that count as an E. So here's a phonetician's E. So that's an E. Now if you say an ah, you'll get a whole bunch of ones down here like that. So that would be, that might be an ah. So we've got the same problem. What we need to do is we need to find the spacing, the spacing between the formants, but we don't actually care exactly where they are. And so you can see that you've got a similar, it's not exactly the same, so there's a few vowels that are distinguished, that are along the same line, but in general, it's the same kind of a problem. And what is the problem? The problem is to somehow detect an E spoken by people with different vocal tract sizes. And the actual frequencies will vary tremendously of the pairs of frequencies, formant 1 and formant 2. So if you have somebody with a small vocal tract, it could move up a lot. And that's why if you just like speed up a recording, without doing fancy Fourier transforms, it will sound like a little teeny little person, because you're effectively shifting all the formants up. So that's how these things might be somewhat similar to each other. You're trying to solve this problem. How do we detect a pair of frequencies? There's actually more than 2 formants, but let's just start off with 2. How do you detect a pair of frequencies, the spacing between them, so that we can determine what vowel it is, and then detect that spacing independent of the absolute pitch, because this direction is basically different vocal tract size, and then this direction is mostly different vowels. So that's like the Doppler shift. So like I said, it's a completely different function, but it's kind of a similar computational problem. So I'll just stop for just 1 second before charging forward. So any questions about, so it's an analogy between the signal processing systems. And so it could be the case that if we look in higher cortical areas in humans, we might find something like this, where you basically have sort of different formants plotted relative to each other on an actual little cortical map, and then summing across the diagonals in order to sort of figure out what vowel is it. So that's just detecting different vowels spoken by people with different vocal tract size. So yeah, there's several layers of things going on. So that's time going that way, frequency going that way. So okay, so everybody clear on all of acoustic phonetics? Not quite all, because we have to talk about consonants. This was like vowels. So what about consonants? Well, these little frequency modulated parts, consonants are kind of like that. And it turns out the BAT is doing a comparison, like the FM frequency modulated 1 dash frequency modulated 2 area is basically doing a comparison between this guy, the time of that guy, and the time of this guy. And what is it doing that for? Well, it's doing that to figure out the distance. So the distance, so the relative velocity is the Doppler, and the distance is the time delay. So it turns out there's an actual map of time delays. So if you look in this FM area, the time delay here is like about 1 millisecond at this end, and that the time delay is like, say 14 milliseconds at this end. And so it's got an actual spatial map of the different time delays between these 2 signals. And what does it use that for? It uses that to try to detect, how far away the insect is, because the Doppler shift just tells it what is the relative velocity, but the Doppler shift, if the insect's far away or close, it's not going to distinguish those 2 from each other. So this is like an example of what a vowel looks like. What do consonants look like? Well, they're somewhat various, but let's just talk about a simple consonant distinction like ta versus da, or t versus d. So if we look at something like a ta, so here's a ta. What does a ta look like? Well, it looks like it's got a little burst in it. So think about how you say ta. So like where does your tongue start? It sort of starts right behind your teeth when you say ta. And it's actually a little bit like an S. Like if you go ta and S, you'll see that your tongue's about in the same place. So there's a little tiny S that comes out, like about, say, 3 kilohertz or something like that. See, that's 3 kilohertz. So there's like a little S that comes out. It doesn't last long, maybe like 30 milliseconds or something like that. And then your voice starts up. But when your voice starts up, what's happening? Well, your tongue, if you look at where your tongue is when you say ah, and where it is when you say just the T part, they're very different. And so the tongue has to sort of start up right behind the teeth. I forgot to put the teeth in, yeah. So there's the teeth, yeah. So it's got to start up right behind the teeth and then move to the ah position. And when it does that, there's going to be, the formant is going to change because the tongue is actually changing the cavities as it's moving. And so what happens when you do that is you get a little transition in the formant before it flattens out. And the other formant is actually going the other way. So you have something like that. So that's over time. This is frequency. So you can see it kind of looks like this kind of signal, and you can actually snip out that little burst, and you can use just the actual shape of these formant transitions to determine what consonant is being spoken. So this is, these are called the formant transition, and what from tongue moving to the ah position. So, and when people have experimented around in bats, what they found was, if I go into this FM area and play if I play something like, I'll do it over here. So if I play something like this into the neuron, it fires. That's all it needs. It just needs like a formant it needs a frequency modulated change and then an echo that comes back a little bit later. That will cause the cell to fire. But if you do this, the cell won't fire. So it's specifically looking for a particular pattern of frequency modulation at 2 different frequencies. And that's kind of similar to what you've got to do to detect these different consonants. You have to sort of detect, oh yeah, there's that particular pattern, and that corresponds to a ta, but if I was going to say a ga, then I would get a pattern that's more like this. And so you actually have, so the BAT processing of these frequency modulated parts has not exactly the same, but it's got a lot of things in common with how you process these consonants, more rapidly changing parts of the speech. And if you look at a different sound like da, like what's the difference between da and ta? Those are basically the same sound. It's the same vowel, but it's actually you're starting in the same place. So think of your tongue, da ta. They're starting in the same place. What's the difference? Well, it's just a delay difference. So what happens with a da is you got a teeny tiny little burst up there at about 3 kilohertz. But the main difference is your voice starts up sooner. So your voice starts up almost immediately instead of a delay. And what are those delays? Well, they're kind of in the BAT range that we're talking about. So over here, the delay between when your voice starts up, it might be like, say, 40 milliseconds. And then here, the delay between that little burst and when the voice starts up might be, 5 milliseconds. And so you can easily detect that difference. Da ta, da ta, da ta. That's the only difference between it. It's just like this tiny 30 millisecond difference in the time of your voice startup. So that's very similar to the bat having to detect these different delays between these different parts of the signal. It's not exactly the same problem, but it's kind of in the same ballpark. It's kind of in the same ballpark, and it's different than the vowel processing. The vowel processing, it's slow and you can sort of average over time. This is looking at these little transient effects that are involving rapidly changing frequencies. So, okay, so that was all of acoustic phonetics in 25 minutes. There's more of it, but this is kind of some of the main points. And so the idea is that, strangely, it might be interesting to look for some of these bat-like processing strategies that are designed to eat bugs to try to understand speech. And so, it's likely to be the case that you might have these higher order maps that are plotting formants against each other, or some maps that are basically sort of plotting a delay of different aspects of the frequency modulated signal, different delays mapped out on the cortex, because you can very clearly see in the bat that there's this whole systematic map of delays, and so it's the cortex is capable of making such a map. So, okay, so I actually didn't go too far over. Any questions? Questions about speech or bats? Or so there's been a lot of other things people have done. They tried to figure out, how does the bat figure out what kind of a thing it's trying to, it's seeing? Is it like a moth flapping its wings, causing other different little disturbances in the signal, or is it a wall, or is it a tree branch? Somehow they can actually detect all these things. And how does it deal with multiple targets? Like it's flying through a room of wires. How the heck does it actually process these tiny echoes coming off all these different wires and actually manage to fly in between them? Yeah, so there's a lot of stuff that's not very well understood. This is kind of like the simplest stuff to decode out of the bat brain, but it might have some relevance to speech. So, is question? Any questions about speech, or the formants, or the tongue, or the pharyngeal cavity, or what the problem is, like this vocal tract size? Because like a small woman will have a much smaller set of vocal tracts than a large guy. So there's quite a big difference there, and an even bigger difference if you look at a kid. The formants are off the chart on a kid with a little tiny kid mouth, but yet you can still understand them. It might be double the frequency. Yeah. Okay, so that's a good question. So, yeah, sort of formant versus harmonic. That's an important point. So in this case, in the bat case, the harmonics, this is just the fundamental frequency of the bat vocal cord. So that's like the bat vocal cords just doing this. And then this is the second harmonic, which is the bat vocal cords, doing the second harmonic. And this is the third. I can't do that with my fingers, but you get the idea. So the equivalent of these bat harmonics are these, the direct equivalent of them are these harmonics of the vocal cord. So this is the vocal cord, doing just the 100 hertz, and this line here is the vocal cord doing the 200 hertz, which goes up to 400 at this end, because I changed the fundamental frequency. So those are the harmonics. So each one of those is an integer multiple of 100. So it's 100. So it's 100. 200, 300. 400. It's just going up. It's just going up integer multiples. But then the formants are the output, of the mouth after you've run all those different frequencies through this filter. So you can think of this as the formant. This is a filter band. So it's filtering out certain frequencies. in particular, if the frequency is, if say this is 500, 1,000. So it's filtering out 1,000. So 1,000 won't get through, but 600 will. So say that's 600. So 600 will. It doesn't matter which harmonic. So if your voice pitch is higher, then the third harmonic is going to get through the filter band. Whereas if your voice pitch is lower, it might be the 1, 2, 3, 4, 5, 6, 7th harmonic that's getting through. So what it is you've got this broadband signal that has all these different frequencies in it, and then you run it through a filter, and the filter stamps out everything except a few emphasized bands. And this is if you look at somebody saying something like you'll see the fundamental frequency of the voice going up and down, but then you'll see those bands, the bands, will be constant. So we're making an analogy between the formants and the harmonics over here. So that's the confusing part. So we're saying that functionally, detecting the spacing between the formants is the same problem that the bats have, but they're actually detecting the spacing between the harmonics. So that's a little confusing part. So it's really like, what causes, the point here is what causes vowels to sound the same when you change your voice pitch. It's just like the timbre of the vowel, and the reason that stays the same is because it's the same frequency. When I go ee, that same band is so like if I did E, if I E, what's going to happen? There'll be all these harmonics going up and down like that, and then the whatever the settings of your tongue are, it's going to say, I just want this, just give me that, and so that's a whole bunch of different harmonics depending on where your voice pitch is. But the functional problem that you have is that these harmonics just shift all over these formants just shift all over the place depending on your vocal tract size, and so you've got to be able to detect the spacing between them independent of the frequency, and that's the same problem over here. You need to detect the spacing between this outgoing and this echo independent of frequency, because if the Doppler shift compensation behavior made the voice higher, well, the whole thing's going to be shifted up. Yeah, it's a little more unfamiliar, sort of talking about these auditory things versus the visual. The visual thing's a little more intuitive. Visually, you're probably not as used to sort of thinking about these auditory processing things. But you're really good at it. Ta-da, 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 ta-da. You can go. Not only can your tongue do it really quickly, but I just went ta-da, ta-da, ta-da. That I did 6 of these things going. It's like a little robot tongue. Okay, any other questions about the speech sounds and bats? Again, one of the reasons to do this is that bats are so good at this, and then so it's easier to see. Humans are really good at it too, but we just generally don't go in there and poke around the same way we do in bats. But that's one of the reasons why people study bats, because they're so good at it. And other animals can do it too, including humans. Like, if you get a blind human, they actually become relatively good at echolocation. They can tell sort of the texture of a wall, how far away from a wall they are. Some really good people can ride a bicycle completely blind, just using echolocation. So so you can become quite good at echolocation if you really need to. So it's not just bats. So this, the same kind of processing strategies, can be used in other animals. It's just the bats are really light and have a really loud, really high voice, and it's really easy. It's easier for them to set these things up. Okay, I'm way over. I think we'll wrap it up there. Okay. I think we're done, David.